My name is KRISHNAN, an assistant professor, PG and Research Department of Microbiology, St.Joseph College, Autonomous, Cuddalore. Today, I am going to give a lecture on antifungal drugs and its classification. Let's start the basic introduction about antifungal drugs. The field of mycology deals with the study of fungi and has resulted in the identification of over 60,000 species of fungi. Fungi are classified as eukaryotic organisms which are primarily classified based on spore production. The classification and identification of a species of fungi by its spore class, in combination with the basic biological mechanisms it uses to sustain life, is key in developing antifungal drugs. The development of antifungal drug focuses on the classes of mycotic diseases for which fungi are responsible. These classes include hypersensitivity, that is allergic reactions based on the presence of mold and spores. Second is mycotoxicosis. So this is based on the presence of fungi that produce toxins in animal feed and human food products. The last is mestismus, that is mushroom poisoning. The disease causing fungi are targeted and then drug classes are classified based on drug structure or mechanisms. Some of the major classes of antifungal drugs include polyene antifungals, azole antifungals, alamines, and echinocandins. It is important to note that antifungal drugs are not limited to these classes, as there are additional drugs capable of targeting fungi that do not fall into these categories. So let's start with the number one classes of antifungal drug, that is called polyene antifungals. So the polyene antifungals are characterized by the presence of multiple conjugated double bonds in the drug structure. So this specific antifungal drug classes targets the fungal cell membrane. The target sterol is ergosterol, so which is specific to fungi cell membrane. In animal cell membrane, it is cholesterol, is a key sterol. The fungal cell membrane becomes leaky, resulting in movement of essential cellular content such as organic molecules and ions out of the cell. Amphotericin B is an example of polyene antifungal drugs. It is selective. Number 2 is called as azole antifungal drugs. Azole antifungal drugs are characterized by the ability to inhibit ergosterol synthesis in fungal membrane. The biosynthesis of ergosterol requires an enzyme called lanosterol 14-alpha demethylase. So this enzyme is needed to convert lanosterol to ergosterol. Targeting this enzyme prevents ergosterol production. So that is the mechanism of azole antifungal drugs. Some of the example of azole antifungal drugs are imidazole, triazole, and thiazole antifungals. Another example for the azole drug is clotrimazole, commonly used to treat athlete's foot, ringworm infection, vaginal yeast infection, and oral thrush. The third type of antifungal drug called alamines antifungals. So alamine antifungals are characterized by the ability to inhibit fungal metabolism. So the biosynthesis of ergosterol requires an enzyme called squalene peroxidase. Squalene peroxidase is responsible for catalyzing the first step in ergosterol biosynthesis. Inhibition of this enzyme results in disruption of ergosterol synthesis. The inhibition of squalene metabolism is toxic to the fungal because of the buildup of squalene and inhibition of ergosterol synthesis. The fourth classification of antifungal drug is echinocandin antifungals. Echinocandins are characterized by their ability to inhibit synthesis of a key component of the fungal cell, while previously discussed drugs target the fungal cell membrane. Cell wall synthesis requires the production of glucose by the enzyme 1,3 beta glucose synthase. Echinocandins specifically inhibit glucose synthesis by targeting that enzyme. So this class of drug is most effective when administered by injection, as it is poorly absorbed when administered orally. An example of echinocandin antifungal drugs called caspofungin. The next classification of antifungal drug is additional antifungal drugs. The major classes of antifungal drugs are discussed above are not the only drug capable of effectively targeting fungi. The emergence of alternative medicine as a hot field of research have also increased the list of available antifungal drugs. For example, numerous compounds and essential oils found in nature have been found to have antifungal property. Example, coconut oil, orange oil, olive leaf, and zinc. The identification of additional antifungal compounds is key to the development of drugs and that can replace existing drugs to which fungi or develop resistance. Thank you.